When will effective life extension technology become a reality? In this conversation, I speak with English biomedical gerontologist Aubrey de Grey, one of the world's foremost experts on the science of life extension. We discuss the root causes of aging, cutting-edge gene therapies, mitochondrial repair, senescent cell clearance, and the large mouse studies Aubrey is currently leading to prove that radical life extension really is possible. This is the Escape Sapiens podcast. If you value these conversations, please subscribe. It's free and it helps the platform survive. And now, here's Aubrey de Grey. I hope you enjoy.
Escape Sapiens.
What is aging, and what's the difference between an old person and a young person, biologically speaking?
I always begin my lectures, especially ones for general audiences, by answering the question, what is aging? Because it's insane. You know, aging has been the number one preoccupation of humanity since the beginning of civilization, and still there's no agreed definition of it. It's ridiculous. And it causes people to talk past each other all the time. So I have a definition that's very simple. It just says aging is the combination of two processes. There's a lifelong process where, literally starting before we're born, where metabolism generates damage. So metabolism is the word that biologists use to denote the entire network of processes that keep us alive from one day to the next. And it causes changes to the molecular and cellular structure and composition of the body. And that's, I'm using the word damage to denote those things. The reason I'm using the word damage, the reason that's appropriate, is because of the second process, which kicks off late in life, and which is where the damage causes decline in function, both mental function and physical function, and that decline is progressive, and eventually it kills us. So basically, the only reason that process is delayed and doesn't kick off until late in life is because the body is built to tolerate a certain amount of those changes without any significant decline in function. And it's only when that threshold level of damage is exceeded that you get problems. So that's all that aging is. And of course, if you think about it, one could define aging of a car or an airplane in exactly the same way. Any machine with moving parts is going to, do damage to itself in the course of its normal operation. And, it doesn't matter whether we call the machine alive or not. So, aging is a phenomenon of physics, not really a phenomenon of biology at all. And I like this definition because it demystifies aging. It allows people to understand that there's no real enigma there, that, we don't need to understand aging better. We just need to bloody get on and do preventative maintenance, same as we do for cars so successfully.
So when you say damage, presumably you also mean damage to DNA, right? And so that makes me wonder, why is it that old people can have young babies?
Okay, yes. So, yes, certainly it includes damage to DNA. And the thing is, we've got tens of trillions of cells in the body. Each of those cells has, most of them anyway, have the same DNA. And that DNA is subject to damage, as you say. So the question then is, what happens in the germline? In other words, in the cells that give rise to babies. And, well, the good news is that there is an enormous amount of machinery that the body has to limit the accumulation of damage to DNA, and indeed damage of other kinds, in the germline. First of all, the cells are quiescent. They don't do much. I'm specifically talking about the female, the oocyte now. Sperm does a lot. It does a lot of work. But the DNA is bundled up really tightly so as to protect it from experiencing too much damage as a result of that work. And then the big thing is, in both cases, both female and male germline, there is an enormous amount of selection. So basically, you end up with the absolute very best, least damaged gametes moving forward. Now, of course, it's fine for there to be a small amount of DNA damage, and indeed it's necessary for evolution, but only a small amount. And so we have evolved enough machinery to make sure that doesn't, that still allows old people to have young babies.
Can you repurpose some of that repair mechanism for the rest of the body, or is that being looked at?
It is being looked at, yes. I mean, there are certain cases where it kind of makes sense. And in fact, DNA is the main thing. So, not very many years ago, a group in Germany discovered something that's now known as the dream complex. And this is a remarkable piece of machinery in the cell that actually suppresses DNA repair. So you end up basically having faster DNA damage than you would if you didn't have the dream complex. Now, the dream complex is turned off in the germline, right? But it's turned on in most somatic cells. And one has to ask why. It seems like a crazy thing. One plausible explanation is that DNA repair slows down DNA replication. When a cell is dividing, it's got to obviously replicate its DNA. And it turns out that you can't replicate and repair DNA at the same time. You've got to get the repair done before you can do the replication. So in early life, in the early embryo, cell division is really fast. And the theory is maybe it's too. The dream complex needs to suppress DNA replication in order to allow that fast cell division to happen. And then evolution couldn't be bothered to turn the dream complex off in cells that are not dividing fast anymore. Okay, that's the DNA. But unfortunately, there are plenty of other types of damage that accumulate in the body during aging. And in many of those cases, the option of repurposing stuff from the germline isn't really available, largely because of the let's call it the life course of the cell in question. So there are cells that are post-mitotic, which means that they're constitutively unable to divide anymore, but they do important jobs. Brain cells, heart cells, muscle cells, and these cells, accumulate waste products, for example. Cells that are regularly dividing don't accumulate waste products because they get diluted out every time a cell divides. But these cells, the brain relies for its function on being made of cells that sit there for a long time without dividing. So yeah, in some cases we might be able to get creative and repurpose things. In some cases, not so much.
Does that fact actually make the brain a very different organ from the rest of the body with respect to longevity? Do you have to do you need very different therapies for the brain as opposed to your heart, say?
No, because of course the heart also is composed of cells that are not dividing, and that
bad choice.
No,
But
no, it's a fine choice because it shows you that actually you don't need to worry. One doesn't need to worry too much about that. That essentially the same processes that could repair damage in the rest of the body will also work in the brain. Well, I mean, of course, the details will always be different from one organ to another, but the brain isn't massively different, except, of course, in one big respect, namely that we can't replace it all in one go. That would rather defeat the object.
right.
Whereas it's pretty likely, I think, that in the early days of truly effective anti-aging medicine, we will end up having some things where we just can't yet do the repair well at a microscopic level, and we have to do like grow whole organs in like a whole new liver, for example, in the lab and transplant it the way we would from a donor. You can't do that with the brain. Though there are some ideas out there for doing it in a kind of piecemeal way in the brain. I personally don't think that those ideas are particularly plausible, at least not yet. But very smart people disagree with me, so it's an open question.
I see. So when it comes to transplant, then say my heart or my liver or something along these lines, is the idea that I would have a donor, someone who's died, their cells would be replaced with mine, and there'd be some telomerase extension or something like that before it's put back into my body. What would it actually look like?
So there are various options that are being explored. One option is simply to grow a whole new organ from nothing, using one's own cells, using the cells of the person who is going to receive the organ in the end. The reason for that, of course, is so as to avoid immune rejection. But it takes time to grow an organ. And also, it's pretty tricky to grow an organ outside the body. People are looking at ways of growing them inside pigs, for example, and this may work, but there are problems there. Then there's the possibility of 3D printing an organ. This has been something that's been looked at for the past 20 years or so, and it's made slow progress. But it's definitely made progress. There are some organs that are harder than others. Some people think that the heart, in particular, will never be amenable to 3D printing simply because the cells need to be joined together mechanically in a very robust way in order to withstand what the heart does. But there may be engineering creativity that will solve that problem. And then the third way is something called decellularization, where you take an organ from a donor, or indeed from a pig, actually, and you bathe it in a rather novel cocktail of acids and stuff, and this causes all the cells to dissolve and go away, but it leaves the extracellular matrix intact. So the extracellular matrix is, of course, this lattice of proteins that holds organs together and gives them their biophysical properties. And in particular, it also defines the vasculature, the blood supply within the organ. That's really the hard part of tissue engineering, is creating a vasculature that is dense enough that the cells that you put into the new organ don't just suffocate and die before they sort themselves out. And this is a very cool way to do it. But again, there are still challenges to making decellularization work for big organs in particular. Essentially, the problem is that when you repopulate, when you recellularize the thing with the cells that from the prospective recipient, those cells have to migrate through this scaffold, and it's hard to get them to migrate very far, basically.
I've seen that we can build ears and things like that, like very thin parts of the body, but it's the larger organs that are a problem.
You've totally got it, yes. Things with a good surface to volume ratio are not so hard. So people have been doing bladders for quite a while. But yeah, solid organs, a lot trickier.
I should have asked before jumping into therapies just about the evolutionary reason for aging. So naively, you might ask, if I were to live longer, I would have a larger chance of passing on my genes. So what am I thinking? How am I thinking wrong here? What actually goes on?
Yeah, so this still gets debated, though not by many people in the field. Most people are pretty happy that that we understand what's going on there. So let me come back to the dream complex. One thing that one might superficially say is, well, this suppression of DNA repair, this is an active process that makes certain types of damage accumulate more rapidly than they otherwise would. This must be an evolutionary choice. This must be because evolution wants us to age. But the explanation I gave you a moment ago is much more the kind of thing that people think is likely to be going on in that case and in many other cases. It's something called antagonistic pleiotropy. Essentially, you've got a gene or a set of genes, a genetic pathway that has a job to do early in life, and it is actively bad for you late in life. But evolution doesn't care about old individuals because old individuals have already passed on their genetic material to the next generation. So evolution doesn't generate or maintain the machinery to keep us alive late in life. Essentially, what we're doing when we're trying to develop therapies against aging is we're trying to augment what evolution has built. Evolution has created this vast arsenal of inbuilt, automatic, ongoing damage repair machinery that accounts for all of the types of damage that the body generates rapidly. Because anything that the body generates rapidly, of course, would kill the organism before the organism had reproduced, right? So it's the things that accumulate, the types of damage that accumulate slowly that are the ones that evolution is complacent about. And our job is to essentially augment that arsenal of machinery that evolution has put in place so as to plug the gaps, so to speak.
So then, one thing in our current society is people are having children later and later in life, right? Women are having children at 35, even 40, and so on. Do you think over time this effect will, lead to evolutionary change where we are living longer just because we're having children later, and that slow damage is going to become more important?
Not really. I mean, or rather, yes, but for a rather different reason, and a reason that doesn't matter. So what has indeed happened, as you say, is that women are making the choice to have their children later, but only a little bit later because they've got this deadline coming up, right? It hasn't changed. Women have menopause at the same age that they ever did. And so, it's not really a big deal right now. Of course, the phenomenon of menopause is linked to the phenomenon of genetic instability. People, and the later you have a kid, the more likely they are to have aneuploidy of some kind, like Down syndrome, for example. Um, uh, Now, medically, of course, there are ways to potentially to fix that. In particular, one thing is that we could actually fix menopause completely, so that a woman's ovaries carried on working and generating new oocytes so that they could remain fertile as long as you like. Now, some women are looking forward to menopause, to not having a period every month, but of course, the idea is one might be able to turn it on and off. So that would give a lot more freedom in that regard. And of course, it would incorporate the safety aspect. The idea is that the genomic instability would be addressed. So yeah, I mean, so one can imagine that as time goes on, if we were to make women permanently fertile, then at some later age, the new problems might emerge that would need to be addressed. But that's the whole point of longevity escape velocity, of course, that new problems will emerge as we solve previous problems, just in the same way that the burden of chronic disease that we have now is really the consequence of our success in stopping people from dying in infancy of tuberculosis or whatever, right? Um, so yeah, so that's likely to happen, but it doesn't. It's not a source of concern because we will address these problems as they arise.
One of the other things that people talk about in this space quite a lot is this distinction between chronological age and biological age, right? Where the idea is that the chronological age is measured by a clock and biological age is measured by markers in the blood or something like this. How scientifically sound is this idea of biological age as opposed to chronological age?
Mm-hmm.
And is it really true that through supplementation you can target, whatever it is in the blood that makes you look like an old person and sort of de-age someone through supplements alone?
Yeah, so the distinction, in order to answer that question, the distinction that one has to start by making is between biological age as an abstract thing and the measurement of biological age. So, sure, there's a perfectly valid concept of biological age. It can be simply defined as the amount of damage you're carrying around in your body, right? That's simple. But then turns out that most of the types of damage that we'd like to measure are difficult to measure in a comprehensive way. And so what we end up measuring is some kind of proxy. Now there are we can classify the types of proxy into two forms. One is functional. We can ask things like, how fast can you walk? How tightly can you grip something? How's your immune response? How's your memory? Things like that. And people have been doing that, of course, for a long, long time. And these things are pretty good. They do tell you they are predictive. Then, of course, there are things that one can do that have also been done for a long time, but that measure something about the composition of the body. Typically, this means measuring things in the blood, as you said. And, the high-end clinics that do this, they'll measure literally 150 things. And again, many of these things change with age, and the faster they change, the faster you're aging. These things are not individually any of them telling the whole story, but they are parts of the story, so to speak. So one can kind of average them, but also more than that, one can get a sense of where one's Achilles heels might be, because everyone's got different Achilles heels, right? So, for example, I've got a gene variant in a gene called MTHFR, which means that I have always had elevated homocysteine, which is a marker for some aspects of oxidative stress and so on. It hasn't done me any harm so far. I seem to have good compensatory genes or whatever to keep that under control, but it's a kind of thing that's worth thinking about. So it makes sense for me to take a particular type of vitamin B12, for example, which is kind of sidesteps the role of this gene. Um,
Can I stop you and ask? Do you actually take this? Is this because what I'm interested in is how much snake oil there is there, and how much is really you trust.
so that's actually the only vitamin, the only thing I take.
Hmm.
And I started taking that fairly recently when the specific cause of my elevated homocysteine was identified by sequencing my genome. Um, in general, I'm the wrong person to ask when it comes to supplements and snake oil and so on. And the reason I am is because overall, all the kinds of things I was mentioning earlier on, I've had them measured half a dozen times at least over the past 20 odd years, and I always come out way younger than my chronological age. So for me, it makes sense to be conservative, if it ain't broke, don't fix it kind of thing, and not to experiment too much. Whereas people who are older than their chronological age probably do need to do something, and indeed, a lot of the things that are available right now may actually work. Whereas if you're already doing really well, there's probably not much additional benefit you can derive.
Yeah, that makes a lot of sense. One
So I
thing
was
that
going to
you
come to the
talk.
final type. I was going to come to the final type of biological age measurement, which is quite important because it's very fashionable these days, and that's omic clocks. So people look at DNA and they look at the RNA and they look at proteins in the circulation or in cells, and they look at things that change with age, and they ask, how much has this thing changed in this particular individual, and they extract a number for biological age from that. And these things are still very much under development. Um, these days, they're pretty good at predicting, your biological age. I mean, in other words, how soon you're going to start to exhibit the chronic health conditions of late life. Um, and they're getting better. But there's an inherent limit here, which is that these clocks are essentially trained on large amounts of data from the population, and that's how they predict biological age in other people. But by definition, the training set consists of people who have not received therapies that haven't been developed yet. So, right. So once you develop a new therapy, you'd like to know that the clock is actually going to tell the truth about that thing, but there's no actual real reason to believe that it will tell the truth, rather than just kind of uncoupling the two things. So, yeah,
I
yeah,
see.
that's a fairly inherent limitation to ohmic clocks.
Yeah, but so can I ask, is the ohmic clock something that you can look at in parallel to the biological clock that you get from, say, blood markers?
Yes, it is. Absolutely.
Okay, so you're interested to see whether those two decouple from one another.
Whether those two decouple from each other, and well, I mean, just in more concrete terms, whether the therapy works. You
Mm.
want to, the point about these clocks is, of all of these kinds, is you want to be able to use them as surrogate markers, surrogate endpoints in a clinical trial. You want to be able to tell more quickly how well the treatment is working, more quickly than waiting 10 or 20 years for somebody to get sick or not get sick.
So one of the things that people talk about a lot, and one of the things that has good science behind it, is this idea of caloric restriction,
Hmm.
right? So you either fast or you take some medication which mimics fasting in your body. And this is shown to give life extension in fruit flies and mice and humans and so on. And so I have sort of two related questions. Why is it that this works? And if it works, why is this not interesting for you? Because I see it's not something that you target in particular.
Yeah. So first of all, why does it work? Well, there is still some debate about this, but there's a very straightforward evolutionary explanation that I'm happy with, and a lot of people are happy with. It goes like this. It just says famines happen in the real world. And anything that happens in the real world, in the environment that evolution has occurred in, is going to be something that evolution wants to optimize for. So if you're in a famine, then reproduction is not something you want to do because if you can have your offspring, but they'll starve to death before they have their own offspring. So it's not going to be evolutionarily effective. Um, so what do? You hunker down. You try to outlive the famine so as to give your offspring a chance. And so this essentially, if we bring that down to something mechanistic, this essentially means reprioritizing how one uses the energy that one has from whatever food one can get, um, away from reproduction and in favor of maintenance, in favor of minimizing the rate of accumulation of damage. And the body does have various choices that it can make. Essentially, a lot of it revolves around growth. If you're not at the top of the food chain, then it's quite important to grow as fast as you can so as to get to a point where you can reproduce before you get eaten. And growth is bad for aging. The faster essentially rapid growth is sloppy growth. There's less time to do the maintenance and and the preemptive damage repair that that could happen otherwise. So yeah, I mean, small dogs live longer than big dogs. Small horses live longer than big horses, and so on. So this is basically why calorie restriction works. That it is simply exposing the animal to something that it has been exposed to ancestrally in the real world, and eliciting the same response that would happen in a regular famine. But here's the bad news, and it answers the second question that you had. Long famines happen less frequently in the real world than short famines. And in particular, the frequency of a particular length of famine is independent of the lifespan of the organisms experiencing the famine, right? So what this adds up to is very straightforward evolutionary logic that says that calorie restriction is going to give less benefit to long-lived species than it does to short-lived species. And of course, this is exactly what we see. We see that we can get nematodes to live several times longer than they normally would if we starve them in the right way at the right time in their life. Fruit flies, at least 3 or 4 times as long. Well, a couple anyway. Mice, 50%. Dogs, 10%. Monkeys, a couple of percent if you're lucky. So this is the main reason why calorie restriction is not the fountain of youth for medical purposes. The same applies, of course, to these drugs that you mentioned, like rapamycin, because what these drugs are doing is that they are activating the same or some subset of the same genetic pathways, what I was calling earlier metabolic reprioritization, that calorie restriction itself does. So there's no way that any calorie restriction mimetic is ever going to do better than what calorie restriction itself does. Now, this is not to say that I think that work on these drugs is worthless, not at all, because, first of all, even a couple of years of extra life is better than nothing, but also because there are health benefits, as long as you do it right. I mean, one could argue that these are health benefits relative to overeating, and certainly, some experiments in the lab, and also for that matter, observations in humans are a bit confounded by that. But in general, calorie restriction and calorie restriction mimetics are a good idea. It's just that one mustn't overinterpret them or think that they are the route to bringing aging under comprehensive control.
I guess also you said it at the start of the conversation. It sounds like you're more interested not in mechanisms for repair that already exist and are evolved, but in new mechanisms that can augment what already exists.
Absolutely right.
Yeah. So, okay, then when it comes to actual damage and repairing that damage, why, another thing you say is as far as I understand your work, you look at repair and rejuvenation as opposing to slowing down aging and the mechanisms that lead to that damage. So why should repair be easier than stopping the damage in the first place?
Yeah, great question. And of course, this was very counterintuitive to the field when I first proposed and propounded it 25 years ago. It took me several years to get the field to take the whole idea seriously. But yeah, I mean, the answer is perfectly simple, really, that damage repair sidesteps our ignorance of how the body works. If you want to slow down the rate at which metabolism generates damage, you've got to take this absolute nightmare of uncommented spaghetti code called metabolism and disentangle it. so as to stop it from doing the thing you don't want it to do, the creation of damage, without having unintended consequences that stop it from doing things we need it to do to keep us alive. And the fact is that's just not possible. The creation of damage is just too inextricably intertwined with the things we need metabolism to be doing. But the point about damage is that it's initially inert. Remember, I mentioned earlier that the process of creation of damage is lifelong, whereas the process whereby damage causes our pathologies of late life only happens late in life. So that means that during most of adulthood, damage is quietly accumulating, harmlessly, inert, right? So if we target that damage, then we're not interfering in metabolism. We're letting metabolism do what it's evolved to do, creating more damage at the natural rate, but still the damage doesn't rise to the level of abundance that kicks off the pathogenesis.
I see. So the analogy would be, so you mentioned cars with rust, right? So the analogy then would be, yeah, we can stop the rust by locking up the car in bubble wrap and in a hermetically sealed environment and never using it. Or we could chip away at the rust once it's accumulated after a few years and still make use of the car.
Yes, exactly. And furthermore, if we do it pretty comprehensively, and of course not just the rust, but also all the other types of damage that a car accumulates in the course of its normal operation, then it lasts indefinitely. The cars that are 100 years old today, vintage cars, they were not designed to last 100 years. They were designed to last 10 or 15 years. And, if you'd asked someone who was building one 100 years ago whether their car would be around 100 years in the future, working just as well as the day it was built, they'd have laughed at you. But of course, if you go to someone who owns such a car now and you say, is it going to be around for another 100 years? They'll say, well, of course, we'll just carry on doing the same preventative maintenance.
So then let's talk about what you're actually doing, right? So as part of the LEV Foundation, the Longevity Escape Velocity Foundation, you're part of these large mice experiments, right? Where you're looking at different therapies and trying to extend the lives of mice. I'm guessing these are the luckiest mice in the lab anywhere in the world, potentially. It's a nice experiment to be a part of, I guess. But so what are you actually doing, right? So what are the therapies? What's the damage that you're targeting, and what can you achieve?
Yeah, so you're quite right. That is what we're doing. If you think about this strategy for damage repair, it's a divide and conquer strategy, right? There are lots of different types of damage that accumulate throughout life. And the only real reason why it's remotely practical to hope to develop a panel of interventions that addresses it all is because these types of damage can be classified into a reasonably manageable number of categories. I normally talk about 7 categories. But even then, it's a lot. There's a lot of things you got to fix. So it all really comes down to one thing that we, the whole field, have increasingly discovered and appreciated over the past 15 years or so, which is that there is a huge amount of crosstalk between these various semi-independent processes of damage creation and accumulation, such that if you fix 1 or 2 types of damage, it kind of relieves the pressure on the body a bit, so that the body is basically better at slowing down the accumulation of other types of damage than it otherwise was. And for this reason, it has been found that if you take middle-aged mice and you do something to them, there are various things you can do that will make the mice live longer, which of course requires essentially all of the types of damage that are accumulating to be accumulating more slowly. And the fact that you can start in middle age is the key, because of course by that time quite a lot of damage has already accumulated. And so, this really means something. And so, historically, at my previous 2 organizations, my focus was on developing the individual components, figuring out ways to repair different types of damage. And my specific focus was on the most challenging types of damage, the ones that it was most difficult to develop fixes for. The reason I did that was because nobody else was doing them. Basically, I've always arranged to be funded by philanthropy, whereas other people, by and large, are funded either by investors who, anyone who wants to make money, they want to make it tomorrow, and or the government, and anyone who's funded by grant applications again needs to get positive results, even if those results are not actually particularly meaningful in the grand scheme of things. So again, it's enormous bias towards the low-hanging fruit. Anyway, so yes, so that's what I've been working on over these past 20 years. But in parallel, the rest of the field has been grinding away on the low-hanging fruit, and the result is that an increasing number of results have been reported by my colleagues around the world showing life extension from various damage repair approaches. So this allows us to move to the second phase of any divide and conquer approach to anything, which is, of course, putting the bits together. Now, in regular engineering fields, that's the easy part. You get the bits working, putting them together, you can be pretty sure that the whole thing is going to work. But in medicine, it's not like that at all, because we're dealing with this system that we have a spectacularly poor understanding of. And so you can get plenty of surprises at the putting together part stage. So I've always been very eager to get that stage moving, and now we have. So yes, you're quite right. Early in 2023, we were able to start the first of these experiments, 1,000 mice, putting 4 interventions together, lots of different treatment groups with different subsets of the interventions. And we were trying to obviously make the mice live longer. And it basically worked. The goal, of course, was to demonstrate additivity. We wanted to take things that individually have shown promise in other people's hands. We wanted, first of all, to recapitulate that, show that we could also do the same thing. And second of all, to get a bigger effect if you had the combination than if you had individual ones. And it did work. We definitely got that additivity. It's that the results were a bit clearer in female mice than they were in male mice, but essentially we got that. And so the only real thing that we didn't get, which we are very eager to get in the future, is a big effect size, an effect size that really, breaks records. To put numbers on this, the mice that we use, a strain called C57 Black 6, they normally live about 2.5 years on average. Mm-hmm. And so we start these interventions when the mice are already 1.5 years old. All right. So they've got a year to live. Now, what has been possible in the past is to get about 4 months of additional life.
So 20%, something like this.
Well, I don't like to talk in percentages, because if you talk about percentages, then you constantly have to be specifying whether you're talking about total lifespan or remaining lifespan starting from when the interventions were begun. So I like to talk in months, right? Okay. So 4 months. Thing is, we could do that 50 years ago with calorie restriction. We could do it 15 years ago with rapamycin, which is the highest, with the best performing calorie restriction mimetic. So we'd like to beat that. The other interventions that we put together in the first experiment had already been shown, as I said, to give some life extension in a couple of months, maybe. But we didn't beat the 4 months. We got to about the 4 months again. I'd like to get to 12 months. I'd like to treble the current world record on this. And I believe it can be done, simply because as time's gone on, even in the past couple of years, there have been more and more reports of new things that actually work, that actually make mice live longer when you give them to them in middle age. And these things ostensibly appear to be addressing different aspects of damage. So you'd expect additivity. They're not going to be just targeting the same thing. So I'm desperate to start the next experiment. I want it to be twice the size. I want it to be 2,000 mice, because I want to do 8 interventions together. And problem is, it costs money. The first study cost $3.5 million, and even then, there are bits that we haven't actually finished yet, tissues that we stored for future analysis. And most of that money is needed right at the beginning, because you've got to buy the mice, and also the interventions, by and large, these are damage repairs, so they're kind of one-shot things that you do at the beginning of the experiment and then you wait. So most of the money is needed at the beginning. For this new experiment, we've identified a few cost savings, but still, there's no way we can bring it down below 5 or 6 million. And, I haven't got the money yet. So I'm working hand over fist every day to try and get it.
doing science outreach.
Yeah.
Well, I hope I can help you out there. But so can I just ask quickly, in your current experiment with the 1,000 mice, did you include rapamycin or not?
We did. Even though it's not damage repair, even though it's a calorie restriction mimetic, we thought, well, we've got to really include it in the first experiment because it's the gold standard. It's the easiest way to, it's the one easy way to get a very substantial increase in life, even starting late in life, starting middle age.
But then, sorry to jump in and butt in. Then wouldn't you expect then to beat the record? Like if you really have additivity and you include the previous record holder.
Yeah. So it's a bit messy. I mean, if you look at the survival curves that I have, been putting out on social media throughout the experiment. So sometimes rapamycin was doing less well, and the combination of all four things was doing better. Most of the time, in fact. So yes, we did we didn't really get quite as much out of rapamycin as some other people have got. And therefore we got additivity. But really what's most important is the situation without the rapamycin. So if you take all the other three things that we did, does the combination of those do better than any of the individual ones? And that, again, was definitely the case.
Okay. So can I ask, so there are three particular therapies or interventions that I'm interested in touching on a little bit deeper. So these are mitochondrial repair, zombie cells or removing senescent cells, I've got a list here, and stem cell therapy. So could we just quickly touch on those? And so to start with, what is mitochondrial repair? And what have you done with mice with regards to mitochondrial repair?
So, so mitochondria are really important components of cells, and they have their own DNA. The only DNA that we have in the cell outside of our regular chromosomes in the nucleus is in the mitochondria. And that's a really bad place for DNA to be, because the main thing that mitochondria do, which is essentially extracting energy from nutrients by breaking them down and combining them with oxygen, that's a really hairy chemical process that creates toxic molecules called free radicals. And so it damages the DNA. Anyway, the cell has, of course, good machinery to minimize the rate at which that DNA is damaged. But still, it accumulates, and people are pretty sure that that damage is bad for us. Though the specific mechanisms for how it's bad for us, or when, or by what pathologies or whatever, it's still a matter of debate. Nevertheless, so we'd like that not to happen. And there's an approach called allotopic expression, which I've been pursuing for 20 years now, which essentially says, let's put copies of the mitochondrial DNA into the nucleus, modified in such a way that they still work, even though the DNA is in the wrong place. And that's really challenging, really, really challenging. Most people gave up on it before I even thought of it. But we've made a lot of progress over the years, much more progress than anyone would have really believed that we could. Nevertheless, we've still got a long way to go. So I've recently been looking hard at other approaches that essentially get rid of mutant mitochondrial DNA through selection, through competition. I would have to get mighty technical to explain the various options there, but the most recent and most promising one may just come down to a single drug, which could essentially alter the selective pros and cons, the evolutionary pressure within the cell, so as to get the non-mutant mitochondria to outcompete the mutant ones. So then you want to talk about zombie cells.
Yeah. What are those?
So the technical term is senescent cells. So it turns out that cells have a whole bunch of different defense mechanisms, especially defenses against becoming cancerous. And one of those defense mechanisms is to lock themselves down into a state where cell division is completely prohibited. But it turns out that cells like that are not just harmless, that they secrete things that are bad for cells around them, and indeed for the entire body. Exactly why this is probably another example of antagonistic pleiotropy the essentially the senescence response has positive roles in the body. The one that's been most explored is wound healing. But sometimes these cells um, don't die when the positive job has been completed. And so, yeah, so the idea is to get rid of these cells so that they don't accumulate and they don't poison the body. The body is pretty good at getting rid of these cells already. The immune system has a role there, but it's not perfect. So some of these cells accumulate. And historically, 25 years ago, when I first started talking about damage repair, I thought that that was going to be quite a challenge. I thought that we were probably going to have to use a rather sophisticated technique that was already routine in the laboratory but had never been used clinically, called suicide gene therapy. But turns out that I was wrong, that actually it's much easier than that, that there are drugs that selectively kill senescent cells. Well, fairly selectively. You have to be a bit careful because they're not 100% selective, but they're pretty good, and they're getting better. So we had a senolytic, as it's called, a drug that selectively kills senescent cells in our first experiment, and we're going to use an even better one in the second experiment. And yeah, I mean, the people who've done work on mice getting rid of senescent cells have shown really spectacular results in terms of widespread benefit to function in these mice. So, you know, this is a big deal right now, and there's lots of startup companies around, you know, doing stuff in that area. And then you want to talk about stem cells. So,
Can I just? I do want to talk about that, but so let's say, for example, I wanted to take one of the current therapies to remove the zombie or senescent cells. What
yep.
would that look like for me as a patient?
Oh, well, I mean, some of these things need to be injected into the circulation. Some of them are orally available. You know, you just eat them. So, you know, this is the nature of these kinds of medicines. They're not, you know, from the point of view of the person who's receiving the medicine, they're not any different than any other medicine. Of course, there's one difference, which is they are preventative in the sense that they are, you know, ideally used when you're not yet sick. Of course, they are. I kind of like to call this whole approach the sweet spot between prevention and treatment, because I just explained why they are prevention, but they're also treatment in the sense that they are restoring the body to something like how it was at a younger age.
And then the next thing was gene therapy, right?
Sure. Right. So a number of the things that we would like to do in the body probably need gene therapy. So I just mentioned senescent cells, zombie cells, as something which I used to think would need gene therapy, and it now looks as though we might not. And similarly for mitochondrial mutation accumulation, that, you know, allotopic expression definitely needs gene therapy. But if this biasing of competition in the cell actually works, then bang, we don't need gene therapy anymore. Well, brilliant, right? So this will continue to happen. There will be times when we can get away without gene therapy when we thought we would need it, but there are times when we're probably definitely going to need it. So a great example is macular degeneration, which is the number one cause of blindness in the elderly. It's caused by the accumulation of a waste product in the cell, in the cells at the back of the eye called the retinal pigmented epithelial cells. This waste product is a kind of reaction product between vitamin A and fatty acids, well, lipids in membranes, and it's a byproduct of the visual cycle. So basically the chemistry of vision, the thing that photoreceptors do when they absorb light. It's a very rare byproduct, and that's why evolution hasn't done anything about it, coming back to what I was saying earlier. But it nevertheless is made and accumulates in these cells. These cells, the photoreceptors, get away with it because they are essentially rejuvenated by the cells behind them, the retinal pigmented epithelial cells, but those cells then accumulate this stuff. And in old age, like 20% of the mass of an RPE cell is this stuff. It's called A2E. So what do we do about it? Well, more than 25 years ago, I came up with a pretty crazy idea, which was to leverage a technology called bioremediation, which is nothing medical at all. It's actually a field within environmental decontamination. So it turns out that a good way to, detoxify a disused airfield that you want to build a housing estate on is to identify bacteria that are able to eat the toxin. So the toxin might be TNT or dioxins or something, right? And it turns out you can do this. Organic molecules that are energy rich, you can find bacteria that will eat them, not because the bacteria want to live a long time, but because they just need energy, and they can extract energy by breaking down the thing. So it works. You can, if you want to decontaminate your airfield, you basically just, identify bacteria in the airfield that are eating this stuff. And the only reason the stuff is still there is because there are not enough of those bacteria. So you get them out, you expand them up in the laboratory, you plaster the airfield with a lot more of these bacteria, and your toxin goes away. Now, my idea was not to inject bacteria into the body. That would have side effects. But rather to identify bacteria that can eat the substance of choice, of interest, so in this case, this thing A2E, then identify the genes that they have that allow them to do so. Typically, there'll be one gene or two, right, that are responsible for this. And then if you've got the gene, you can do gene therapy. You can you can stick that gene into human cells, and the human cells can suddenly break down this stuff that they were previously unable to break down. So this is the plan. And it took a long time to get it to work, but it did. It was the topic of one of the startup companies that we spun out maybe a decade ago from my previous organization. And they've continued to progress with it, and now it's in the process of being sold to big pharma. So it's quite a promising approach for addressing macular degeneration. And the same thing can, in principle, be done for various other waste products, like oxidized cholesterol, which is the main driver of atherosclerosis, for example.
Mm-hmm. It sounds like there should be a lot of money for your experiments if things go right.
If things go right. So this is the cross I have to bear, is that I work on the earliest stages of these research projects, and everybody knows, yes, that they will be extraordinarily valuable in the future. But the paradoxical thing about medical research is that even though each stage of the research costs a lot more than the previous stage, if you go through from cell culture to mice to phase 1 trials to phase 2 trials, phase 3. Even though they cost a lot more as time goes on, nevertheless, it's actually easier to raise the money for the later stages simply because of the de-risking that has happened. Every stage you succeed in, there's a greater chance that you will finally make it to the finish line. So yeah, I'm at the sharp end of having to fund the earliest stages, which are the hardest to fund, even though the actual dollar amounts are quite modest.
So then let's say I was a billionaire and I was going to throw around $1 billion. Why should I invest in you as opposed to any of the other competition, let's call it?
Well, first of all, let's not use the word invest, because my organization is a non-profit. So you give me money, you're not going to get it back, right?
Well, if I want my life to be longer, let's say.
Well, that's true. But actually, it's more than that. So there are many people in this field who are very much investors, emotionally, rather than donors. They don't like giving money away. But many of those people have now invested in longevity companies, actual for-profit companies. Now, of course, they want return on that investment, and that depends on demand, right? So ultimately, the faster the overall longevity industry grows, the better their return on investment, right? So it actually makes very good, rational, mercenary sense to give me money so as to accelerate the rate at which the whole field grows, and the rate at which people start to realize that aging is within striking distance of being brought under comprehensive medical control.
It's like a multiplication factor on whatever other investments they've made in other areas. I see. Okay, yeah. So then what does the blueprint and timeline look like? So in terms of longevity, escape velocity, what does the future actually look like moving forward from today?
So I, of course, always answer such questions probabilistically. I do not know how long it's going to take to get to a particular point, but I feel it's my duty to give my best guess of how soon we have a 50% chance of getting to a particular point. So there are 2 points that I always highlight, two thresholds, two milestones. The first one is this 12-month increase in mouse lifespan with treatments begun in middle age, so trebling of what we can do today. I call that robust mouse rejuvenation. And the second one is longevity escape velocity, the point where in humans we are able to postpone the health problems of late life by long enough that we buy ourselves time to figure out what to do next and so on. And I believe that long enough is about 20 years. If we can postpone aging by 20 years, then we're going to be at longevity escape velocity, and we're going to be able to
And that means long enough for you personally, or?
Oh, no, no, because I haven't given any time frames yet, right? So I believe that we've got at least a 50% chance of getting to robust mass rejuvenation in the next 3 or 4 years, just so long as I can raise the money to do these experiments. In fact, probably more than 50%, to be honest. And I believe that the impact of making this dramatic breakthrough relative to what we can do today, and it has already been able to do for 50 years, will completely invigorate the field. What I want to do, and this is my new slogan, I want to make aging the new COVID. I want to get the world as determined to bring aging under medical control as it was 5 years ago for COVID. And I believe that the even though the public don't care about mice, the subject matter experts do, and the subject matter experts are very influential. You know, we go on Rogan all the time. We go on, Oprah and Lex Fridman and whatever, because they're already very interested. And if we can start saying more optimistic things and actually giving predictions of timeframes, then it'll be a different world. Okay, so I believe that that's what's going to happen. There's going to be a proper war on aging triggered by getting to robust mass rejuvenation. And I think there's a 50% chance that that will get us to longevity escape velocity within another decade from that point. So, in other words, 12 to 15 years from now, something like that. Now, most people say that's completely crazy. It takes like 30 years to get one drug through the FDA. But, of course, that's the beauty of COVID. That's why COVID is the best thing that's happened to the longevity movement this decade. What we saw was that, when humanity decided to put its finger out, everything we thought we knew about regulatory inertia and bureaucracy and so on was just swept away overnight. And we got vaccines both developed and distributed globally, literally 10 times faster than had ever been done before. So, yeah.
In a post-aging society, let's say people can now live to 500, 1,000 years, what do you think births, deaths, and marriages are going to look like?
So I don't really like to look at the distant future very much because so much is reliant on unknown unknowns, especially things about the development of other technologies, unrelated ones like, automation. So honestly, I have no idea. The focus that I have is a slightly different one. My focus is not on how society will be when people are living vastly longer than they do today, but rather on the very much nearer term situation when people start to expect to live a lot longer than what they do today. Because that's what's going to happen. If I'm right about just getting to robust mass rejuvenation, triggering a proper war on aging, people are going to be thinking that there's a good chance that they themselves will benefit in terms of never getting sick when they get old, right? And therefore that they expect to live a long time, even though nobody yet has actually lived a long time, right? And what does that mean? I mean, it means huge things. If you think about the big drivers, the big ticket items in the global economy, most of them revolve around how long people think they're going to live. Think about life insurance, health insurance, pensions, inheritance arrangements. Everything is going to be upended. So whenever I give talks to, investment banks or, pension funds or whatever, the talk I give tends to have a title along the lines of anticipate the anticipation. I say, it's your job to look at a world in which people are expecting to live a lot longer and to figure out what your product range ought to be in that situation so that you can roll it out when that expectation materializes. And the reason you should be doing it now is because when that expectation does materialize, it's going to happen essentially overnight. There's going to be an absolute sea change very, very suddenly when, the influences of this world line up and demand a war on aging, and people start talking about it the way I always talk about it, but the way hardly anybody else does.
There's this great expression, or this expression from, I think it was Planck, where he says that science advances one funeral at a time.
Yeah.
Right? I wonder, do you think that if we live longer, there'll be an impact on scientific advancement? So
yeah,
that,
maybe. First of all, that's a problem I'd like to have. But secondly, it's trivial to fix. There's a two-word answer. It's called term limits. I mean, you can give tax incentives to switch careers. Anything. It's trivial.
I see. I know that you don't like fear mongering, like dictators are going to live forever and so on. But let me.
It's not so much I don't like those things. It's more a case of they're just so ridiculously easy to rebut. And let's take that one. Let's take dictators living forever. Last I checked, dictator was fairly high on the league table of risky jobs. Not a lot of dictators die of aging in the first place. And the ones that do have tended to organize their succession beforehand, so they're functionally immortal already. Come on.
I suppose the fear that people have is that, much like you want to remove senescent cells from the body, maybe we want to remove the dead wood for the human body, for the human species, let's say. These are the sorts of arguments that people make, right?
Right. And like I said, you can remove them to different careers or even different places. So Elon Musk comes out occasionally and says things like that. Says like, you're going to, aging is a good thing because otherwise we would have this cognitive ossification. And I say, well, it's okay. We can just ship all the old people to Mars.
Let me end then on some advice, right? So I'm not going to ask you about what the average person should do to make sure they can take advantage of longevity, escape velocity, eat well, and so on. But there are two things I'm curious for your answer on, and that is, should we be storing tissue as a reference for later in life? When I was a child, I had a tooth removed, and I think that still exists somewhere in my mother's house or somewhere, probably. And I know that the enamel is very protective and so on. So maybe that's a good reference for when I'm 80 years old and want to take advantage of some therapy. Is this a recommendation you'd make?
Yeah, not really, to be honest. I think it does make sense to store, baby's cord blood and so on, but not for reasons of aging. I think these are mainly for reasons of, low risk, but non-zero risk, early onset, medical problems that kids might have, where the stem cells might be useful. The reason I don't think it really makes sense for aging is simply because we do reconstruct. We can reconstruct pretty much whatever information we want about how someone was at a younger age. Old people even still have plenty of perfectly young, but functionally young stem cells. It's just they don't have as many of them as young adults had. So really, plus, of course, as time goes on, we're getting better and better at these things. So, if someone is 20 now, for example, and they store some tissue, it's going to be 30 years before they have any aspects of aging pathology. So, that's a long time in science, by which time we won't really need it.
So, final question. Should we all sign up to Alcor for cryogenic preservation?
Well, I've been signed up with Alcor for 23 years. I certainly think it makes sense. Partly because we don't know how long everything else is going to take. You remember, I always give my time frame predictions as probabilities. But secondly, it's for different people, right? I think I'm fundamentally in this business for humanitarian reasons. I don't get out of bed in the morning because I want to, live longer myself. I mean, I do want to live longer myself, but I know that my work will only very slightly improve my own personal probability of making it to longevity, escape velocity. Whereas every day that I bring forward the defeat of aging, I'm saving 110,000 lives. That's quite easy to get out of bed for. But of course, however, whatever the timeframes are end up being in practice, there will still be people for whom it's too late. And those are the people that we need to cryopreserve. Now, of course, the problem at the moment is that cryopreservation is still very much a research area. In other words, the damage that is done to the body by the cryopreservation process is still very substantial. So that even once we are in a position to address the medical problems that caused this or that person to be pronounced legally dead in the first place, if we can't fix the damage that was done by cryopreservation, it's all a bit academic. So there are a number of ideas out there for improving this, for dramatically reducing the amount of damage done by cryopreservation. But it's even more pitifully underfunded than my work, and that's saying something.
Aubrey, thanks for coming on the podcast, and I do hope you get the $5 million you need for the next study.
Well, don't just hope.